Dr. Gehlenborg already said, my talk is titled Visual Exploration for Cancer Subtype Analysis, and this is about a paper that was published in a visualization conference called Euroviz last summer, and I have the reference at the back of on the last slide. So, first of all, I want to acknowledge the team of people who are working on this project. It's a fairly large number of collaborators that are working on this project with me at Harvard School of Engineering and Applied Sciences, at the University of Linz in Austria, the Graz University of Technology in Austria, and then at Harvard Medical School. And we are also working very closely with the cancer program at the Broad Institute and their Tcga, the Cancer Genome Atlas Genome Data Analysis Center there. So, I want to start with a question. Why do we want to do cancer subtype analysis? And the answer is surprisingly simple. It turns out that it's quite important, because knowing about cancer subtypes allows us to treat patients better and ultimately come up with better outcomes for these patients, and also to develop more targeted drugs in the end. So, how could we go about identifying subtypes in cancer? And I already mentioned the Cancer Genome Atlas project. That is a very large-scale project funded by NCI and the National Human Genome Research Institute in the United States. There are about 20 centers that are generating a lot of data for a lot of cancer patients and cancer samples. So this project is studying around 20 cancer types, and the goal is to have roughly 500 patients at least per tumor type, which I know you can do the math yourself, but just in case you can't this morning, that means there are about 10,000 patients, and for these 10,000 patients, we have a lot of data. So we have, among other things, we have mRNA expression data, we have microRNA expression data, we have DNA methylation data, we have protein expression data, we have copy number variation or copy number variant information, we have mutation calls for them, and we also have a lot of clinical parameters. So that's actually a very rich clinical data set, even though it has a couple of holes. So, with all of this data, we can do stratification, right? So we could take this mRNA expression data and we can cluster our patients, and we'll end up with a number of clusters of the set of patients. And so for everything that I'm going to say from now on would refer to one of these tumor subtypes. So it's not 10,000 patients, but it's 500 patients. Okay, we can do clustering. We can stratify patients, for instance, based on the copy number or mutation status of a gene. And whatever we do, when we stratify our patients, we end up with a couple of groups of patients. And once we have those, we can ask, well, so now we found a cluster, for instance, by clustering mRNA expression data, and we can try to find correlations between those clusters and say certain gene mutations or copy number changes or some clinical variables and so on and so forth. We can also ask whether there are different outcomes. Say, do patients in one of these clusters live longer, or are they maybe younger people or older people? And so there are many, many questions that we can ask, and here are just some examples. And now the question is, how do people typically do this once they have their clusters and they want to correlate these different data types with each other? And I just want to show you from this last paper that I had up earlier. This is by Verhaak et al. Came out in Cancer Cell in 2010, which was sort of one of a key study that used Tcga data to identify tumor subtypes in glioblastoma. And this is a figure straight from the paper. This is how they represent their subtype information. So basically the top, it's proneural, neural, classical, mesenchymal, those are the final results. Those are the 4 subtypes that they identified, primarily based on expression data. But they also then correlated these 4 subtypes with various other bits of information. Whoa. Oh. Okay, so for instance here on the top row you have a mutation status of TP53 in those patients, IDH1 mutation status, and you can see right away, for instance, IDH1 mutations are more prevalent in this proneural group than in the other 3 groups. There's only one, in fact, that is not in the proneural group, and that's in the neural group. You can see gene expression profiles here. You see copy number levels for genes where it says CN and so on and so forth. So the idea is simply to create a matrix to group the patients, which are the columns here, based on the subtype, and then line this up with various other molecular and clinical information. Okay, and so they're clearly not the only ones that are doing that. So here's a picture that I sneakily took at a Tcga symposium last fall. So here's another example. There's a heat map, and then we have all these different clusters and different subtype assignments up here. I know it's very blurry, but just to give you an idea, here's the clustering heat map. Here are some subtypes, and then all the other information is lined up there. And you can see this doesn't really scale very well. Here's another example where we have, again, a heat map, and at the top we have all these different other variables that the analysts were interested in trying to see if there are any correlations between those. Okay, so since this doesn't really scale that well, and this approach that I've shown you with these heat maps and just lining up different categorical variables in the end above the heat map doesn't really scale. So we've developed an exploratory tool called Stratomex, which in fact stands for Stratome Explorer, and Stratome is maybe a badomics word. We made that up. Maybe we'll actually win that award that's given out for horrible omics words. So Stratome is supposed to represent the set of all patient stratifications that you can have in a data set. And as I mentioned earlier, there's a really, you have almost endless possibilities. You could just look at binary mutations status of any gene, and you can stratify your patients based on that. You can look at many different clustering algorithms, and all of those will give you different stratifications of the patients. And we're trying to explore that space and identify overlapping clusters in the end, and clusters that have interesting outcomes or associations. So what does this look like? This is a screenshot of the software. And what we have here is a gene expression heat map that was clustered into 4 clusters. And I know it's very small, so I'll explain it to you. So each block in here, in this column, represents a cluster. You've probably figured that one out already. The columns are the genes in this case, and the rows are the patients. Okay, so this was just a random clustering result that I picked. Now, we can add more data to this. And so, you hover the mouse above this area up here at the top, you'll get a plus button. You click on the plus button, and you get this little wizard that allows you to choose what you want to do next. And you can basically do 2 different things. You can either add a new stratification to this view, which would be the top button, or you can add a dependent column to that. So some variable that you want to group based on this existing stratification. I'll show you some examples later. So I'm going to select a new stratification, and then again I have a couple of options. So this is like a menu, and you drill down. So we can cancel at any time. You can also go back up 1 level. And essentially what I have here is a list of options to query this database of stratifications that I loaded into the software. And so the first choice is simply to pick a stratification from a list. And I'll show you this in a second. So, I chose that to pick it from a list, and I get a new window down here, which is called the tour guide. So this thing down here will guide you through the Stratome. And you probably can't really see it that well, so I'll make it a little bit larger. And basically what you have here is a list of clustering results on methylation data. So red is methylation data in this data set. And we have basically taken a DNA methylation matrix, so it's basically computed based on genes and their methylation status. And we have clusters ranging from 2, or clusterings ranging from 2 clusters to 8, and we also have sort of an ungrouped group in there. And we can then sort of add additional data types, microRNA or mRNA, and our list is getting longer and longer. And so this gray one here is actually the one I had selected earlier. That's why it's sort of highlighted. You can see the number of elements in these stratifications, because we don't have all data types for all patients in most cases. So sometimes you'll only have like 100 patients for a data type. In other cases, you have 500 or more than that, the number of groups. And this additional column here shows you basically distribution of patients in those groups in the clustering. So you see in this case the two cluster versions are pretty evenly split in most cases, but this one, for instance, is not. Okay, so yeah, I'm adding another data type, but I think you get the idea. And then I just resize the window again, and then I clicked on one. So I selected a hierarchical clustering with 4 clusters, and then I immediately got a preview over here. So you can see it's a preview because the cancel button is still on there and so on and so forth. It has a box around it to show me that I'm in preview mode. And well, the other clustering that I had loaded was a consensus non-negative matrix factorization clustering. And we basically are comparing 2 clustering algorithms on the same data set in this case. And these bands here, they represent patients that are that are basically contained in the intersection between these 2 sets on both sides, right? So we see this cluster number 1 has very large overlap. Basically all patients in cluster number 1 are in cluster number 1 in the other clustering. But we can also see that that's not always the case. And so once we confirm this here, we can also select clusters and see where those patients end up in the other clustering. We can do this here as well, and we see, for instance, this cluster is almost evenly split into 2 in this clustering. And finally, what we see as well, very easily, this 4th cluster in this hierarchical clustering really is like, it probably contains 4 patients or something. So it's not surprising that they don't line up very well. On the other hand, this is a very nice way to explore how your clustering algorithms perform and to compare them to each other. So we can also see a selection of the patients, their IDs basically that are in those bands. We can select individual bands if you don't want to select the whole cluster, basically selecting the intersection between 2 sets. And then, because they don't really look that great, so there's not a lot of overlap between these 2 4 cluster versions on the same data set. So let's just try to make a query. Instead of just going through a list, we're now going to actually compute some similarity between clusterings and try to find one that matches this. So we're going to select, again, that we want to add a stratification, but instead of selecting it from a list, we're going to do a query that is going to return clusterings that match this one over here. And to compute the similarity, we're using the adjusted rand index. And it's very straightforward. So this thing will tell you what to do. In this case, it tells you click on a header brick here, or a header block. So we clicked on this one, and then the tour guide will immediately show us the results of this calculation. And we got a new column here that shows us the similarity score. And I think, again, I'm going to make this a little bit bigger. So the wider the bar, the better the overlap. And that's essentially a 3-cluster version, again, with CNMF. That is sort of the best result for that, and we can, and again, just select this, and or we already got it in the preview mode here, and yeah, we can now compare the clusters here, and we see they still are not perfect, but there's pretty good overlap between those clusters in general. So this cluster number 2 apparently is the one that's split when we go from 3 to 4 clusters in this clustering algorithm. Down here, I just added a little bit of detail. So we have a little histogram in here because you can never really see the distribution of the scores very well. So you can view that as well. Then I've taken out this mRNA data type from the database, and then this thing will automatically recompute the query, and then we get another result here, which actually looks pretty horrible. But according to the adjusted rand index, this would be the second or the best result if we don't look at mRNA. So this would be looking across 2 data types. And when you look at cancer data, generally you don't see a lot of overlap between really clusterings in 2 different data types. That's why we can also do something more interesting. And in this case, so I'm actually just removing these sort of sample results here. That's very easy as well. And what I'm going to do now is I'm going to do a query on a group. So again, I'm going to go in here. I want to say I want to look for another stratification. And this time I'm going to search based on the overlap between one of the clusters in here and whatever we have in our database. So I'm going to select this thing down here, and I select cluster number 2. And now what I'm going to do is I'm going to look in the gene mutation list. So I've deselected all the other data types and only selected mutations here at the bottom. And you can see the stratifications are now basically genes. So the name of the stratification is the gene symbol. And then we get additional columns that show us how many what percentage of patients has a mutation in that gene and what percentage doesn't have a mutation in that gene. So we can see this immediately without loading anything in here. And then we can select results from here. For instance, in this case, I selected EGFR. And again, I immediately see, okay, so there are mostly patients that don't have a mutation in EGFR, but quite a substantial number of patients do have a mutation in EGFR. In this, in fact, I should have mentioned that. That's the glioblastoma data set from Tcga. So we can look at this and we see actually there's an orange line around this upper block here, which means that the query matched this non-mutated group. So since we're actually really interested in genes that are mutated and overlap with clusters, we can simply go into our database and deselect the not mutated status, and then the tour guide will again update the query, and we see the top hit is now EGFR, and the hit is on this mutated group. Okay, so let's see. Okay, so I then confirmed this cluster. I want to keep this one. And now we can do queries on this group, for instance. So one interesting query or question to ask when looking at gene mutations in cancer is to see if they're mutually exclusive mutations. And what we can do is we can again add a new stratification. We select the cluster overlap query that's based actually on the Jacquard index. That's simply the overlap between two sets. We select this as the query, and well, the first hit in this case is Titan, which is a huge gene. So if you're sort of blindly searching for genes that have mutations, you'll get this a lot. But that's nothing to worry about. In this case, you see P10 is the next result, and we see that this is a really good match. And there is, in this case, this is not based on mutual exclusivity. So, and the reason why that was is because I had deactivated this not mutated group, so I couldn't even find anything that is not mutated where EGFR is mutated. But when I deselect this, I find NF1 as a gene that is indeed almost perfectly mutually exclusive in terms of its mutations in the patients. Okay, so now I'm going to do something potentially crazy because I have 2 minutes and I just wanted to give you a very, very brief live demo. Here we go. So this is the software. I'm just going to throw in a stratification from the list. I'm just going to select this. So here is the clustering from this Verhaak paper applied to a subset of the data. I confirm this. I can now throw in, for instance, a pathway. So let's see how this data looks like when we map it on the glioma pathway. So I'm just going to type in a little search query. We can compute, we can find pathways using gene set enrichment analysis on these clusterings, but for that I would have to load the whole matrix with all 24,000 genes, which I didn't do in this demo data set. Okay, so now we have the pathway here, and I just realized we need to scale this a little bit because of this projector. The resolution is not great, so here we go. Maybe a little bit small for the people in the back of the room, but so what I can do now is I can also, open these things up, and this is a histogram, so I can just, add a little more contrast to this, and maybe you can see this now. I don't know. I apologize. This is a challenge with visualization software and projectors, but we can zoom in here, and you can see the genes that we have in this matrix. These are the 1,500 most variable genes out of this expression data set. They are now mapped to this glioma pathway, and you see, for instance, here, this is actually PEGFR, and EGFR is here at the top. So you see it's downregulated in this case. When we open up another one down here, let's see if I can get this done. We see EGFR is actually upregulated in this case. And now we can ask, okay, why is this? And I'm instead of doing a query, I'm simply going to show you why that is. So I'm just going to select from the list. I'm going to select the copy number data down here. And I know we're looking right now interested in EGFR. So I type in EGFR. We see, oh, EGFR actually has a lot of increased copy number levels. So there are mostly low level and high level copy number amplifications. And this cluster here consists basically only out of patients that have either a high level or a low level amplification, mostly high level amplifications. And what I said before Well, this is the cluster where we see that EGFR is highly upregulated. So this could have something to do with that. And before I get kicked off stage, I just want to show you another thing that we can do with the software, and that is to look at outcomes. So, well, maybe we can just do this very simply like this. So we have clinical variables. I just selected I want to add a clinical variable, a numerical clinical variable. We have a couple of those. Let's see if I can. I'm not going to do this. So right now I have age. I have age at time of diagnosis, days to death, which is basically survival, and we have days to last follow-up in this case. But I'm just selecting days to death in this case, and I get a set of Kaplan-Meier plots here. I can again open this up. I can mouse over here, and we can see, okay, there's one group that seems to have slightly better survival, and it is this proneural group, which is the one here at the top. And we can now, I can try to do this. I don't know if this is actually going to work that well. I can add another gene. Again, I'm simply going to add it from the list. I want to look at the mutation status of IDH1, which is also known to play an important role in this tumor type, or which is one of the genes that characterizes these subtypes that were found in the Verhaak paper. So I confirm this. I see there are not that many patients really. But then when I look at clinical data here, oops, sorry, I made a mistake. I want to actually make this clinical variable dependent on this gene mutation. So I just select this from the list, and then if I look at days to death here, I see there's actually a significant difference in survival between these two groups, at least a notable one. So that's maybe as a comment here, we're not trying to compute any statistics. We're loading everything into the software. We're getting the data from a Tcga analysis pipeline that basically processes all the Tcga data automatically. That's a system called Firehose that's running at the Broad Institute. And we actually have this all pre-packaged. So if I just go back to my presentation, you'll see this. When you we download the software, I'll show you the URL in a second. There's a tab, Tcga data. You see this pipeline runs every month, or it was running every month. Now it's going to run every 3 months. You have basically all the tumor types in there. If there's a tumor type that you're interested in, you just go in there, select it. It'll download the data package, and you'll basically get all these different data types, the clinical data, and so on and so forth. And you can play around and explore this. This StratoMax software is implemented in a framework called Kaleido. It's a visualization system for molecular biology data. You'll find it on this website. You'll find it on GitHub. It's completely open. It's under a BSD license, so you can take it and sell it if you have customers. And down here is the citation.
All right, thanks a lot. We have time for a couple of questions. If you have questions, please speak up. I don't see microphones here in the hall. Yes.
So the software is for free, is that correct?
Yes, yes, it's open source software.
So can you add your own data to this?
Yes, absolutely. So the reason why we're putting all the Tcga data, or we're able to preload this more or less, is because it's a large data set. So it would take a while for you to do this, but you can easily load in any matrix that you have. We have a very nice import dialog where you can load in a matrix, can select the columns that you want. You can pick mappings between identifiers. Say if your patient identifiers are not the same in all your data files, which happens a lot in Tcga data. If you want to map the different gene symbols, you can do this in the import dialog. You can assign data types, whether it's a categorical variable, whether it's a ratio, and so on and so forth.
Can I run it on the server?
No, this is a desktop application. Yeah.
How about filtering samples by, say, alias outliers? Can I remove samples from matrices?
You can split clusters, so you can basically select a band, an intersection, and say you want to split off that group from the rest. You cannot really take out samples from the data set at the moment. Yeah.
Is it Mac software or is it cross-platform?
It's cross-platform. It's a Java application. It's implemented as part of within the Eclipse Rich Client platform. This is the same framework that Eclipse is using. So we can build binaries, more or less, that you can deploy on basically any popular platform, and they're all available on the website. So if you want to play around with this right now.
Question back there.
So any facilities for automating analysis? So very often you do something, feel with someone you want to do a comparison against or
Right. No, we don't have that. Since our goal is to get the human into the loop to make sense out of the data, so we don't want to automate anything. So this is really complementary to analysis pipelines.
I mean, again, like there's this tension, right? You want to automate something, you want to human. They say that the vision is to remove automation.
Sorry, say it again?
So your vision is never introduce automation into the process?
Well, we're trying to leave automation out of exploratory analyses.
So, but there would be, it's effective to have a loop. You have a pre-analysis, you have your intervention by the human, you have a reanalysis, you have an intervention, but it seems that feedback to the analysis is not realized. Is that right? Not at this point.
At this point, we have not realized this. What we have is, since the idea is to find sets of patients, groups of patients that share something, you can take any of these clusters and either export a list of patients in that cluster, or you can export the patients and the data for that cluster. And the idea is that you would export this, and maybe you can reanalyze or analyze further using R. But we're currently not directly tied in with anything. The problem is also, of course, that for those data sets, you need an analysis pipeline. Typically, these firehose runs take several days. So you need something fairly powerful to follow this up. But this is something we're definitely aware of, and we're planning to do this in the future to make this easier to sort of iterate over this process. Absolutely, yeah.
Okay, thanks a lot. We have 3 minutes break now to